So good evening. It gives me great pleasure to welcome you all to the 2016 Grant Ingram Lecture with our distinguished speaker, Professor Sally Otto. I'm Chris Lee. I'm the acting principal of St. John's College, and I would like to begin this evening by acknowledging our presence on the traditional, unceded, and ancestral territories of the Musqueam peoples. So the Grant Ingram Lecture is one of the most important events here at St. John's College. It is named after the late Grant Ingram, our founding principal. Dr. Ingram was a distinguished physical oceanographer who held many leadership roles here at UBC. And this lecture series was established in his honor to carry on conversations about the topics that he was passionate about, including First Nations communities, science and the environment, and the Canadian North. Dr. Ingram was the late husband of Dr. Helen Burt, who is currently UBC's Vice President for Research International Pro Tem. And we're really delighted that Helen has taken time from her busy schedule to join us tonight. So thank you, Helen. So the final thing I will do is to introduce our keynote speaker tonight, Dr. Sally Otto, who is professor in the Department of Zoology at UBC and director of the Biodiversity Research Center. Dr. Otto received her PhD from Stanford, and her areas of research include studying population genetics and evolutionary biology. Using mathematical modeling and experimental evolution, she investigates the evolutionary forces that have generated the remarkable diversity of life. Now, it will take me all the way to dessert to just scratch the surface of what Dr. Otto has accomplished, but let me just list several of her many awards, including a MacArthur Fellowship, a Guggenheim Fellowship, the McDowell Award for Excellence in Research from UBC, a Stacey Fellowship from the National Sciences and Research Council, a Stacey Prize from the National Research Council of Canada, and the Sewall Wright Award from the American Society of Naturalists. And just a few weeks ago, Dr. Otto was named a Killam University professor here at UBC, which is the highest honor for faculty here at UBC. So please join me in welcoming the 2016 Grant Ingram Distinguished Speaker, Professor Sally Otto.
It's really a pleasure to be here, and I really appreciate all of you coming. I wanted to give this talk because the genetic technology is changing so rapidly, and I feel that there are many decisions that we have to talk about and that we have to decide about where our comfort levels are. And if we don't know about them, then we're not going to be able to get to decide. So I do think that the more we talk and the more public awareness there are of these novel changes, the more informed a decision we can make as a society. So I'm really, really grateful to my panel co-members, Sally Aitken and Emily Drummond. They're associates of mine in the Biodiversity Research Center, as well as Daniel Steele, who's at the Maurice Young Center for Applied Ethics. So thank you. As Chris mentioned, these are some of the topics are sticky, but I do think we need to talk about them. So I'm going to have a two-part introduction. I want to set the stage for your thinking and for the panel. The first part is what are the challenges and what is the scope of the challenges that I think we are facing. So I'm a biodiversity scientist, and I'm going to give you a somewhat biased perspective of what, as a biodiversity scientist, do I see as the great challenges facing our planet. So what do I mean, first of all, by biodiversity? In this talk, I just mean the wealth of biological diversity that lives on this planet. And there are an estimated 5 million different species, and these 5 million different species are like treasure troves of the history of evolution since in the 4.1 billion years that life has been evolving on this planet. So we see all walks of life on this planet, and it is what makes our experience so wealthy, so rich when we are outside and experiencing the natural world. But this biodiversity is increasingly in crisis. And to give you a sense of the extent of this crisis, 1 in 5 species of vertebrates, animals, invertebrates and vertebrates, and plants are at risk of extinction right now, according to the IUCN. 1 in 5. And this extinction risk is much higher. It's been estimated at about 1,000-fold higher than background or natural levels of extinction risk. Just to put some faces on these abstract numbers, here are Canadian species at risk. So, for example, the little brown bat, the bank swallow, and the eulachon, that's culturally quite important in the Fraser Valley. It's a very important First Nations these have all declined over 90% in my lifespan. So 90% declines in these particular species. In general, across vertebrate species, in general, across my lifespan, there's been about a 50% decline in the population size sustaining these species. So those are dramatic declines in numbers of the wildlife that sustains our natural spaces. And I think these numbers are even going to be worse because some of the challenges we haven't yet seen. We've created a world that's warming, an ocean that's acidifying, but the worst of those changes are yet to be experienced. Nevertheless, work from UBC, including Sally Aitken, who's on our panel, and Tong Li Wang, has shown that there is, with a warming climate, we're seeing greater and greater challenges for the wildlife that lives in British Columbia, and there's a mismatch between where species are found and what's the best climate for them. And you can ask, how fast is that mismatch expanding? And I think this is a remarkable number. They found that it's at 1,000 meters per year. That's the mismatch is increasing at that scale due to climate change. 1 kilometer a year. As a result, people are watching and seeing our species keeping up, and some are, but many, many are not. There was a recent study showing that bumblebees are failing to keep up, and trees are failing to keep up as the climate warms and the places that are best for a species are shifting. So we could talk all night about the fundamental drivers of this biodiversity crisis, but I do want to spend a little bit of time about what I think are the ultimate drivers, and that is the 3 problems of humanity: overconsumption, overpopulation, and overuse of resources. Overconsumption, a term from UBC as well, from Bill Rees, the ecological footprint. One can quantify how much could the Earth sustain. That is, you could use this amount of resources, but you could grow that amount in a year. So that would be 1 Earth. Here in Canada and the United States, we consume many, many more resources than that, and the estimate has been made that we would take 5 Earths to sustain our level of consumption. 5 Earths. We only have 1. And across the world, there's huge inequities in the consumption rate, but across the world, it is over 1. So we are living beyond what could be sustained in the long term by this planet, and the the footprint is getting larger over time. In addition to overconsumption, we have overpopulation, which is a big part of the net footprint as well. There are 7.4 billion people on this Earth, but that number is expected to increase by 50% by the end of the century. So somehow we have a biodiversity crisis now, but I think if we're going to feed and populate this world with another 50%, we're going to face even larger crises. And it's not just how much we consume and how many of us are consuming, but how we consume that matters. Because we can consume in ways that recycle a lot and that have very low impact and try and concentrate their impact on a small part of the Earth, or we can try and consume everywhere. And that's kind of what we're doing. And a study that kind of accentuates this point from IRES, again here at UBC, in Navin Ramankuti's group, has tried to categorize how much of the world have humans affected. What's rangeland, what's in forest land or cropland, and what's wildland. And I think the wildland numbers are pretty dramatic. They estimated that less than a quarter of the Earth's total land space, if you exclude the ice-covered parts of the land, is wild. Less than a quarter. So humans have dramatically sculpted the planet. And I should say that they were looking in fairly coarse areas, 86 square kilometers. So they were asking, is it pristine, is it intact or not? And less than a quarter of our planet is intact and pristine. So I focused on the biodiversity crises facing the world, but I think a lot of the other crises facing the world are all tangled up in this and affected by these same 3 root causes: overconsumption, overpopulation, and overuse of resources are also at the root of conflicts across the globe over resources, whether those resources are oil, water, forest, fish. And there was even a CBC report this week about sand. If it's a resource, we're fighting over it. And of course, as exemplified in the ecological footprint, the resource use is not equitable across the planet. There are people that have a lot of access to resources, that the only way we can live on 5 planets here in Canada and the United States is by importing the resources and consuming down the resources, not only in our own countries, but in other countries. We'll also see, as the population increases by another 50%, a severe toll of malnutrition. Already, 1 in 9 people across the globe do not have enough food. We need to feed another 50% over the next century. And finally, another big challenge clearly facing the globe is how do we tackle diseases? And you wouldn't necessarily think that that's related to overconsumption, overpopulation, and overuse of resources. But in fact, the emergence of new diseases and their spread is highly related to the population density. A disease can't spread if there's no other people to infect. And so by urbanization and increasing density, many diseases that would not be able to spread, would not be able to get a toehold in the human population, can now do so. And the graph to the right just shows the increasing emergence of new diseases over the last 60 years. So overconsumption, overpopulation, and overuse of resources have led us to a situation where we're living on what I would say is a fragile planet. Whether we're looking at biodiversity or the emergence of human diseases, there are many crises facing our planet and humanity. And I think that we are all facing this moral imperative, a moral challenge. What do we do? How can we bring the Earth back from this very fragile, very precarious state to one that's sustainable and robust, and that can sustain the other species around us as well as humanity? And of course, I think that these being the big drivers, I think that that's really where we have to spend a lot of time. But we are going to be increasingly called upon to use whatever tools we can to solve these challenges. And so what I want to do now is shift from, this is my perspective, and it's idiosyncratic, but this is my perspective on the challenges facing the Earth. And I want to now turn to one area of solutions and techniques that people are going to be offering and that we need to discuss as to whether or not we want to use those challenges, use those technical solutions. So I want to talk about the potentials and risks of using genetic technology to tackle the great challenges. The reason why genetic technology has been changing so much is really we just have so much more information, so much more data. And this graph shows just since 1985 what the growth in our databases, our main databases on genetic information. And this is on a log scale, so it's just been increasing astronomically. And when we say whole genomes, whose genomes are we talking about? More and more, we're filling in our understanding of the genetics of this tree of life with over 3,000 animals and plants and over 74,000 bacteria, where we have information about the genes throughout their genome. So this knowledge revolution is leading to a technological revolution. And I want to talk about an array of different technologies that this is enabling. The first few technologies are more kind of traditional technologies where we can use genomics to inform breeding. Humans have been breeding animals and plants and domesticating them for 12,000 some years. So that's what I mean by the more traditional methods of crossing and preserving certain offspring over others. So one example from UBC and Emily Drummond's lab, led by Lauren Riesberg, has been looking at what are the genes in sunflowers that make sunflowers that we eat and the sunflower oil that we consume come from a flower like this when the wild relative looks like that. So this has changed that humans have selected, domesticated over the last few thousand years, and they can pinpoint where in the genome these genetic differences are. And that can be useful because sometimes we actually don't want traits that are in our domesticated strain, but we might want traits that are in the wild relatives. So here's a really dramatic example of that. These are strains that have been bred with a wild relative, and they're trying to winnow down the segment that's coming from the wild relative, but they're looking for genes or segments that are present already in our wild relatives but allow the crop to survive in locally challenging conditions. They were looking in this case for drought tolerance, and this is an example of the results from the field study where they have in Uganda really quite severe drought, and you can see here these are some plants that have a segment inserted or bred in from the wild relatives right by other strains that are dead. So you can see the potential here for tapping into the genetic diversity that exists in the wild relatives of crops to try and improve agriculture and improve the ability to grow productively in more places across the world. Another example comes from Sally's lab, where she and her colleagues have been looking at what allows or has allowed trees to grow where they grow in either very cold climates or in warmer climates. And in a study that they just had published in Science, they were able to identify several of these regions, pinpoint where they are in the genome, that have allowed trees in some places to survive in very, very cold weather, and in other places to not be able to live under those conditions. So they found hundreds of gene regions in pine and in spruce that allowed temperature tolerance, as well as tolerance to different latitudes and different elevations. Why does this matter? Well, knowing better what genes can enable a tree to survive in a changing climate means that Sally and her group are trying to help improve our reforestation strategy. When we cut down trees in British Columbia, we reseed. But where do we take those seed from? Normally in a pretty close region to where we cut the tree down. But this can help improve our reseeding strategy to say, well, the climate that will be faced in the future would be better matched by seeds of trees in these other locations, potentially greatly improving not only productivity of our forestry industry here in British Columbia, but also potentially saving some species from going extinct if they fail to match the moving climate. But now I want to turn to really what is kind of a trickier issue or a more novel aspect of genetic technology that has emerged in the last few years. And this has also come from genomics and kind of opening up this knowledge that we didn't know about before. And one of the most shocking discoveries has been that bacteria have immune systems, adaptive immune systems. Nobody ever suspected that. It's called CRISPR. It works entirely differently from our immune system. But the way it works is really, really cool. It was known for a while that there were these repeated arrays within the genome of bacteria, many bacteria across both archaea and eubacteria. But they didn't know what it was, what those little segments were. As more and more genetic data became available, they found out what they were. Little segments of viruses or little segments of other foreign DNA that had gotten into the cell. And the bacteria had a way of storing them, cutting them and inserting them into the genome. And then in future points in time, those little segments in this array, this CRISPR array, would be expressed and sliced and put on this little Cas9 carrier that went around the cell looking for something that matched. And if this cell got recently attacked by another virus that matched the one in its repertoire, it would cut it up. Cas9 would go and cut it up. So this is a remarkable way of remembering, storing information. Bacteria are storing information about past assaults and past foreign DNA that has gotten into the cell to help them combat future attacks by viruses and plasmids. So this is an immune system in bacteria. Super cool. Very fundamental science, but it has completely revolutionized genetic engineering. And Sally and I get like these flyers in the mail and email every day talking about how CRISPR can change our research. So how is, let me connect the dots between this funky immune system in bacteria and genetic engineering. And to do that, I want to step back just a couple steps and talk about what's the more traditional genetic engineering that biologists have been using for the last 20-some years. So one example is in plants. Plants, and genetic engineering is often done in, there's lots and lots and lots of different ways you can do genetic engineering. Like any engineering, there's lots of different methods. Here's one. It piggybacks on the natural genetic engineer. Agrobacterium tumefaciens has a circular piece of DNA. It inserts into the plant genome of its host, and it manipulates that plant genome to give it more resources. And this forms galls. So what humans do is say, okay, let's stick in some DNA that we want to go into the plant, inside this plasmid, and we put it in the cell, and it'll get inserted somewhere. But therein lies the problem. First of all, it doesn't always go in. Second of all, you don't know where it's going to go in. And third of all, because you don't know where that gene is going to go in, you have to build this construct to have all of the components of the things you need. And sometimes that can who knows what you're disrupting, and who knows what the potential costs and trade-offs are. So it doesn't always work. It's hard to do. It's very hard to do. In some organisms, it just doesn't work. So what about CRISPR? CRISPR is changing genetic engineering because, at least in principle, in any organism, if you can get the gene in that makes this Cas9 scissors, then you can insert where you want to cut. This is a template piece of RNA that says, I want to cut something that looks like this. Just like the bacteria said, I want to cut anything that looks like this past disease. So then that goes through the cell, and it finds in the host genome something that matches. Cas9 says, oh, got to cut there. So now you've got to cut. Well, the trick of genetic engineering with CRISPR-Cas9 is not only do you put that in, but you also put in a DNA strand that's what you want to go in. That matches on the sides, and it recognizes, oh, this is a really similar piece of DNA. It matches on the sides. I'll copy it in here using the way that organisms like to copy in DNA into cut points. And that transforms this from whatever it was before to whatever you want. So now this type of technology, just inserting changes, allows us to be much more precise. Because now genetic engineering can say, this is the exact site in the genome we want to cut, and this is the exact replacement we want to make. And some of those replacements will be quite benign. For example, in the drought case, rather than going through many, many generations of breeding, maybe if we know exactly which base pair or change in the gene is present in the wild relatives, then this could be cut in this kind of surgical way without many, many, many years of developing that new crop. So that's one possible application. So remember this is CRISPR is an editor. It can be used as an editor to change parts of the genome in a much more precise way. But now where I really want to make sure that the discussion is opened up is that this has also been used to as the basis of what's called gene drive. So CRISPR technology is not all about gene drive, but I'm not going to change and talk about what CRISPR with gene drive is. And fundamentally, the difference is that not only are you editing, but you're putting in the editor. Not only are you doing genetic engineering, but you're putting in the genes that can genetically engineer. You could make a mosquito that carries a gene that sterilizes a good fraction of its offspring, or that skews the sex ratio to males. Male mosquitoes don't bite, they don't transmit disease, and potentially, if we drove all of the population to males, we wouldn't have mosquitoes. And the ethical consequences of that, I think we need to talk about. Other cases where I think that we will be called to consider gene drive are what if there's an invasive species that we know is eating all of the eggs of an endangered bird, or that is crowding out a plant. Should we use this technology to cause that invasive species to go locally extinct, to drive its population to only males and then to flip out? For agriculture, should we use it in the pests and maybe make them less interested in eating our raspberries or strawberries, or potentially, again, eliminating them? So you can see why I wanted to have this conversation. This technology, using CRISPR for gene drive, is a technology that has only been talked about in the scientific literature for the last 3 years. If you do a search of the Canadian website, the canada.ca, there is not a single document on gene drive about how it should be used in research or released, other than what is already encompassed by our regulations for genetic engineering in general. But as I've mentioned, the scientific and the ethical issues are much more complicated because of this rapid wildfire spread that this type of gene drive can do. So I just want you to think about the types of risks that we might want to talk about. I should also say that, of course, scientists are also trying to figure out ways to reduce or reverse gene drives that do spread afoul. But risks that I think are really important to think about is, what if we wanted to get rid of an invasive species in Canada, but it was native in Australia? And it would not necessarily take much traffic between Canada and Australia for that, for specimens to get there and knock it out outside of where we wanted it knocked out. It could spread beyond the target genes. Many species hybridize with others, so we could intend it for one, but it could jump to another species. Another example is, we may think we know what the target is, and we might think we know what the replacement is, but mutations happen all the time. Rearrangements happen all of the time. So it's not under our control what happens in nature. And maybe other elements will move in or mutate, and so we can have consequences not of what we intended. Another possibility is that resistance will evolve, so that we spend all our money doing this rather than other ways of protecting our planet or protecting the species, and it doesn't work because resistance evolves. And because it's so young, we have no idea how fast that will be and whether it'll be fast enough given the time scale of the spread. And ethically, I am not comfortable about driving a species to extinction, even if it's a mosquito. So, and I know people feel differently, but what is reasonable for humans to do? And then the other thing, and I don't want to overly scare you because you got to eat, is that this technology is really doable in a lot of different labs and in a lot of different places on Earth. And it's not necessarily always going to be used benevolently. So that is a scary, who gets to decide? It may not be who we want. And as I mentioned, the guidelines right now don't exist in Canada. We need to ask our government to move quickly to address it, to have public conversations, to talk about these issues, and to regulate accordingly. Esvelt, who does a lot of gene-drive research, has argued that regulation will be too slow. Genetic technology is too fast. These changes are much on a faster time scale than policymaking. And if that's so, does that take the decision away from the public? Who's deciding then? Should it just be scientists, or how do we open these kind of questions up so that we as a society can choose what the right balance is? Thank you.
So thank you, Sally, for that fascinating and in many ways very discomforting discussion of what's possible. But this is what St. John's is for. So I invite you all to enjoy the dinner. We will convene after dinner with our panel. And for table hosts, please feel free to encourage the discussion. So I'll leave it up to you now. Thank you. So let me begin by just briefly introducing our panelists. They'll be joining Sally for this conversation. First, we have Dr. Sally Aitken, who's professor in the Department of Forest and Conservation Sciences, and she's the director of the Center for Forest Conservation Genetics here at UBC. She is the project leader of AdaptTree, a large-scale study using population genomic techniques to understand local adaptation to climate in conifers and to better match genotypes with new climates. I think we learned a little bit about your research, so we'll be looking forward to learning more during this conversation. We also have tonight Emily Drummond, a senior PhD student in Dr. Lauren Reisberg's Sunflower Ecology, Evolution, and Genomics Lab. We also learned a bit about your research earlier as well. She completed her MSc in Zoology here at UBC and her BSc at the University of Toronto. Emily has worked in many systems, from bacteria to flies to tropical plants, and her current research focuses on agricultural weeds and their evolution. And we're also glad to have Dr. Daniel Steele, an associate professor in the School of Population and Public Health and at the W. Morris Young Center for Applied Ethics. Dr. Steele's research focuses on coupled ethical epistemic issues in science, especially in relation to environmental and public health issues. He's the author of multiple books and articles, and in leading journals and on the philosophy of science. So please join me in welcoming our panelists tonight. And I believe Sally will start our conversation off, and then we will also be inviting you to pose questions. So if you have a question, raise your hand, we'll spot you, and at some point we'll integrate the questions to our panelists too. So please.
I think I just wanted to say that we really want to have this panel open to your questions, because it's really about having a conversation, and I can imagine that some of this is new, a lot of it is new, and so pose any question that you may have. And the other thing that I think is important to state for myself, but I think it's true for the whole panel, is we don't know either. We don't have, it's very hard to figure out the answers to these large scale questions. So, Sally, do you have any words that you'd like to add before we open up to questions?
Well, I think one of the really challenging things about this is just how complex it is and how difficult it is to have a conversation that involves complex science but has great implications for the public. And Sally did a fabulous job of explaining some really complicated stuff in ways that we could understand. It's a much tougher thing to do that to a broad general audience, and especially in today's very short attention spans on topics. So it's important we're having this discussion, but I think that with many things that we're dealing with, it's very easy to fall into a yes or no, good or bad kind of dichotomy, and these issues there's just so much nuance involved that it's very challenging.
Emily, did you want to add your thoughts?
Yeah, sure, I will. Thanks again for inviting me, Sally. I'm filling in for Dr. Reisberg, my supervisor, and big shoes to fill, but certainly I've worked in his lab a long time and thought deeply about these issues. And it is very timely because the rate that the technology is changing at is quite astounding. You know, we are in the genomics age, and this has some really big implications for how we go forward. And so one thing I wanted to point out is that the August issue of National Geographic, if people are interested in learning more about this, maybe some people have seen already, has a big feature on CRISPR and gene drive, and it has lots of examples and kind of explanation of how scientists are thinking of applying this technology. So I just thought it was kind of coincidental, but people might want to check that out.
Thank you. Daniel.
Yeah, well, I don't know if I have that much to add. Maybe what I was thinking is to sort of prod questions by getting us to think again about the first scenario of the three that was on the table, the one about the mosquitoes and the gene drive technology. And I think there are just, as a philosopher, when I see an example like that, one of the things I think about is there's a lot of frames, backgrounds you could use to think about this kind of example. It could be, well, maybe this is like pesticides. Well, if it's like pesticides, well, then what does that tell us? Or is it really a new thing that's fundamentally unlike some previous ways of controlling unwanted pests like mosquitoes and other things? And also other examples of attempts to control life forms that we didn't like. In some cases, like eliminating, say, smallpox. Well, that seems really good. Extirpating wolves. Well, at the time, wolves probably seemed as bad, to people, settlers in North America, as mosquitoes might seem to us today. In retrospect, extirpating wolves may not have seemed like such a good idea, and some of the reasons might be similar to reasons that getting rid of mosquitoes might not be such a good idea, in terms of their role in, say, an ecological network and so on. So, I guess my thought is, your questions don't have to be about that one, but maybe that's a place to start, that scenario. Mm-hmm. If people have questions about Sally's question. Well, getting rid of a species, isn't there just something ethically not right about that? Even if it's a really bad species like mosquitoes that kill millions of people, or
So would this be a good time to open up for some questions from the
that
floor?
would be great.
Great.
Thank
So
you,
please
Chris.
put up your hand if you have a question, and we'll need to ask you to shout a little louder so we can hear the front. So please.
And you can ask clarification questions before we get into the scenarios, or just launch straight into the scenarios and the kind of ethical dilemmas that they raise.
Yes, please, go ahead.
I was just curious, whether you know how this is being used right now. How has it been used in very concrete ways
Right.
so far?
So far, it's in the lab only. And it is, so with the gene drive rather than the CRISPR, the gene drive rather than the CRISPR has only been done in the lab and only been done in a few species. For Drosophila, they're starting to work in mosquitoes, and I can't remember the other two, but they were both insects.
No plants.
Yeah. But that's why I also do think that that's going to be the first question to arise. Is it's going to be about malaria, it's going to be about dengue and Zika.
So one of the points in the first narrative is that we don't know how it's going to manifest. So is there a way of testing it in the lab to predict the ways in which it will manifest?
I think that predictive modeling is a very tricky thing, and any model is built on some range of parameters and some range of situations or scenarios, and I think even with, mostly we work. Tongli here works on modeling future conditions for different plant species with climate change. And that's just leaving something like CRISPR, where you've got a self-perpetuating molecule introduced. And there's so much uncertainty. There's always a great deal. So I think that if somebody said they could predict exactly what could happen, I think that you'd have to view that with considerable suspicion. However, you could model a wide range of scenarios from best case to worst case and get some idea of what might happen if things go very well and what might happen if they go very badly. So modeling certainly could be a useful tool in that regard. But we have to be very careful about those unexpected events that aren't built into the model. You're a modeler.
Yeah, I think that's exactly right. And some examples are, in the lab, you could find out how stable is this construct over a few generations. You can answer that. You're not going to be able to answer what is the risk of hybridization, because that could be a 1 in a million, 1 in a billion event, and so it depends on how many mosquitoes are affected and where they're living. So that's not testable in the lab. So there's things that really will not be testable in the lab.
We have a question in the back.
Well, I'm just curious. It's like maybe we are sure that mosquitoes are not important for human race, but are we sure about that? Like they have no environmental importance at all. They don't play any kind of role in the environment, and because extinction is an important thing. It's a big step.
Yeah, so I think you're right in saying that from a very human-centric kind of view, mosquitoes have no importance and they're a pest, and we would feel a lot better if we got rid of them. But you're right that from the perspective of the ecological community, that is not the case. Like, lots of things eat mosquitoes, rely on them as a food source, for example. So that is completely unknown what would happen if we eliminated mosquitoes in certain areas. What would happen to the species that perhaps depend upon them? Would they be able to find an alternative food source? Would they not? Would we have some kind of cascading effect of this extinction? We kind of don't know. I mean, in some senses, this is a big experiment we're already unintentionally running with the mass extinction events that Sally Otto is talking about. You know, we're starting to find out how communities might change as you start losing species and how unpredictable that is. So I think that is maybe a factor, you know, in any decision we make. Do we have the right to kind of change a whole community this way? Yeah.
Yeah, in response to that, one of the members at our table was suggesting that we simply raise mosquitoes in the lab, male mosquitoes, and let them out to substitute for the natural population that we have driven to extinction. And since there would only be male mosquitoes let out, their role in the environment or the ecological network would remain unchanged, whereas their potential for disease spreading would have been zero.
Well, that would be a big lab to make enough mosquitoes. But I think the point is a good one, that safeguards are really important. If this is done, then we can think about what are safeguards and keeping a breeding population of mosquitoes if things go wrong. But as Emily's saying, it might not be the species, you know, you don't know what the downstream consequences. Well, you have saved and bred in the lab all of the aquatic insects that eat mosquitoes. You won't have necessarily done that. I did want to also add, that's why it's also nuanced in that some of the gene drives that are being proposed for mosquitoes don't get rid of mosquitoes. They just make it not possible for the mosquito to transmit malaria. And so is that going to be ethically acceptable more? Certainly it's not going to have as many of the ecological unknown side consequences, but it might drive Plasmodium extinct. Maybe that disease agent, maybe that's, you know, where maybe you're okay with, maybe people are okay with that, not maybe okay with mosquitoes. I don't know where those answers lie, and that's what Daniel was saying is hindsight. You can learn the ecological impacts by hindsight.
Yeah, but there's also an ethical question there. I mean, so in the examples of these species eliminations, I mean, I guess like smallpox is a virus. So that might be in a different class than even the Plasmodium. But you know, is eliminating a microbe, eliminating a mosquito, eliminating a wolf? The wolves? Okay, so at least nowadays, I'm sure 100 years ago, people were like, get rid of those damn wolves, right? But I mean, nowadays, probably most of us, especially here at BC, would say, you know, killing all the wolves is bad. We probably think that. I mean, it's okay if you don't think that, but that would probably be, you know, these kinds of. But okay, mosquito, well, maybe, I don't know. I mean, do people, and this is throwing this out, do people feel like there's a difference between wiping out a mosquito, eliminating a mosquito that's a vector for a disease, or killing the microbe that's the thing that gets carried by the mosquito that makes people sick, and eliminating that? I mean, my own intuition is, is that, yeah, I mean, if we could, if you could somehow snap my fingers and make Plasmodium, the microbe that causes malaria, disappear forever, I mean, I don't know. I think if I could do that, I would, get a grant to do that, and my lab did that. I might win a Nobel Prize for that, right? So I mean, I would be personally, I mean, I might want to think about maybe ecological consequences, but I would be okay with that. But I don't know. Do other people have questions or?
Yeah. My question was about the previous answer. I was going to raise a question that you wouldn't necessarily have to wipe out mosquitoes, wipe out malaria. I think inherently there is a difference between targeting Plasmodium and targeting mosquito. Largely answer my question.
There's a question in the back. Yes, ma'am, second. Go ahead. Yes, please.
My question is that related to the issue of right to exist, that is species. Who has the right to exist in this organism? We often take a anthropocentric viewpoint that whatever would contribute to the human well-being, they would have the right to exist, directly or indirectly. Now, given your scientific analysis and discovery till now, does there exist any species on the earth which you think doesn't have the right to exist because it has zero contribution? And if the ecosystem is indecomposable, you cannot decompose it into parts, then the answer is very difficult. You cannot make a marginal separative analysis that yes, this is the marginal contribution of this species, the species A, species B. It is possibly the totality of the complex of species that really contribute to even what humans consider as bad. What is your?
As an evolutionary biologist, my view is that every species alive today has the same evolutionary amount of time that its lineages have existed on Earth. We're all equal in that evolutionary sense. So it is very hard to say that a Plasmodium doesn't have any, well, there's no rights in that viewpoint. It's just an evolutionary viewpoint. But I think there's another issue that is raised here, and that is everybody has a different view. They come with a different cultural perspective, a different moral perspective. So everybody in this room is going to have a different answer and for different reasons to that question. So how do we decide?
First of all, thank you for doing this panel. Couldn't agree with you more. This seems highly relevant and an issue we need to discuss. I mean, the consensus we came to at our table was generally this seems like the 21st century equivalent of rabbits in Australia. But I'm interested to get your thoughts on using gene drive as a tool for potential astrobiological applications. Now, I know that's a whole other kind of goldfish, but say in a biosphere setting to sort of use it for, yes, anthropogenic uses, but it seems like somewhere where the application could have minimal negative side effects, sort of on Mars, let's say, for example. After Elon Musk unveiled this week.
That kind of goes back, I think where you're going is similar to the other question, is can we experiment somewhere safe? And I think this is interesting because you're like, Mars might be safe. I think if we have to go to that level of containment.
Not to do it.
But the other thing is we can't even answer that, right? It's like the lab that doesn't have the same risks because the risks of hybridization and jumping species aren't there. So yeah, I don't know if that's exactly where you were going, but I think that is a way. It does raise a containment solution.
Right. Well, coming back to Earth, people have long kind of thought of islands as almost more self-contained units. And so some of the more interesting cases, at least to me, are cases like Pacific Islands, where not as an intentional consequence always, but because of humans arriving, we've brought other guests along with us that were never there to begin with that have completely decimated the native fauna. So things like in Hawaii, where we're losing all these bird species to basically like a bird malaria that came along with whalers quite a few decades ago, or things where we've brought in predators that bird species also have no experience with. So maybe that would be one case, as Sally pointed out in her talk, things might still escape from the islands, so you would want to think about it carefully. But a case of, say, trying to undo what we already did might be one scenario I could think of.
Daniel, and then we'll go back to the corner after. Daniel?
Yes, so this is actually an extension of the earlier question where in the case of smallpox, which is a virus, and mosquitoes is a vector, the question is, what kind of a slippery slope are we going to get from here? Now we say that we're going to get rid of mosquitoes to stop Zika and dengue and the sort. But are we going to get rid of bats to stop rabies? Get rid of rats to stop plague? And where does that end?
Daniel, you want to?
Yeah, I mean, that's in some ways, so yeah, I mean, that in part before contemplating doing such a thing, those are obviously questions, tough ethical questions to ask, yeah. And I don't know all the answers to that. I don't think anyone knows all the answers to that, or I'm not sure there are answers to that in the way that there are, true or false answers to lots of things. But yeah, I mean, that's the part of what these scenarios are. The first scenario is getting to think about. It's like, well, what value do you put on a species that we normally think of is really a really bad thing, in which, in fact, we devote a lot of resources to trying to kill, right? I mean, through other means, right? Through insecticides, through draining swamps, through, you have to mow your lawn, because if you don't, anyway, you can't leave standing water, and all kinds of resources we devote to not having these things around and getting rid of them. Well, what if we had some way to get rid of them? Now, in part, there's a science fiction aspect to this. The conceit that, yes, we can get rid of them. And that's, I know when the four of us met for coffee this week, and that was, for me, taking this bigger look at these kinds of issues, so from a historical and philosophical kind of view, when these new things, pesticides come along, these new things come along, just, this is a new big thing. This time we'll get rid of these things once and for all. And usually nature is a little more complicated and tough, and especially these kinds of bugs are a lot harder to get rid of than we think. So I don't know, maybe this case is different, but I have a grain of skepticism that we really could get rid of the mosquitoes, even if that's what we really wanted to do. Even if we decided we want to do it, could gene drive really wipe out some strain of mosquitoes? I mean, I think it's, again, when we talked about scientific uncertainties a lot at our table, and to me that's one of the scientific uncertainties to keep in mind. But yes, certainly before contemplating doing such a thing, you'd want to think about what are the values of these different species, ecological value. Do species just have an inherent value in their own right, even if they're species we don't like? And we think over history, the kinds of species that people like and don't like really change a lot, like the wolf example. So it's, I don't know, 100 years from now, people who would consider themselves enlightened look back, how could they be so mean to those poor mosquitoes? We have a mosquito reintroduction, programs in our national parks and things like that. So, who knows, right?
So this is going back to a comment made earlier regarding, like, instead of wiping out mosquitoes, maybe you should treat the Plasmodium instead, and come up with better treatments for that. But what about vaccinology then? Do you think it would be more valuable in the human context to focus on making vaccines to prevent the infection rather than trying to wipe out a species that is a vector for that disease?
Do we have any virologists or in the room? People who work on vaccines in the audience?
Well, certainly there's a lot of research being, this is, I would say, a really small fraction of the research being done on how to prevent the spread of malaria and how to cure it. I don't know anything about vaccine development and what's holding that up for malaria. Maybe somebody in the audience does.
I know a bit. I actually was just reading that the Bill and Melinda Gates Foundation has funded malaria vaccine development with GlaxoSmithKline.
Yeah.
So this is kind of what I started doing my undergraduate degree a little bit, and so that's why I was kind of
Gene drive?
Gene drive, there we go.
Yeah. So, and I think there are a lot of subtleties. So the thing about a vaccine, again, is creatures evolve really quickly. And so there's a lot of diseases for which a vaccine is not very effective and not very effective for long. But the same is true potentially for gene drive. And as an example, so one of the thoughts about how, let's say we wanted to protect a population from a gene drive that's being used elsewhere. So one suggestion is maybe you can drive through first in that population a segment that cannot be cut by that same scissors, right? And so you basically immunize that population, and then you do your gene drive where you're replacing it with your element that would drive that species extinct or whatever. But then that's a way in which a natural population, if it already has things that cannot be cut, that's one of the kinds of scenarios that Daniel's talking about, that the real world may not be as susceptible to driving extinct. The species are really robust. There's a lot of evolution that's constantly happening, but there's also a lot of things that go wrong.
I have a Todd and then two Chief and then John Henry. Okay. I study history of communications, and I was picking up on buzzwords such as audiences, public, conversation. So I'm just wondering, how do you imagine this conversation unfolding beyond this room, beyond the scientific community, even beyond national borders?
That's a really important question, and I think that conversation is maybe happening elsewhere better than it is happening in Canada, and so right to the Canadian Geographic. Talk to your parents. Talk to your neighbors about these issues. I would love to see this conversation spread.
If I, sorry, could I just follow up on that? So one of the, I think that we can take some lessons, looking back in history, I think we can take some lessons from what happened with genetic engineering in the 90s, where there wasn't a public conversation. And really a lot of things were happening behind closed doors, and it was scientists and regulators, scientists who were advocating for the technologies that they had developed and or were working with, and regulators who were very sympathetic to them. And we ended up, what that took us to is a situation where there's a lot of the strong sentiment against genetically engineered crops isn't actually about the crops. It's about all of the ownership of germplasm, the control over that, the control over the food supply, and just the fact that back when there was no public dialogue on it. And I think this is something that Sally brought up in our conversation earlier this week, that we need to avoid the situation where the scientists that have, or companies or whatever, who have the most to gain from the technology are the ones driving the conversation.
Well, this kind of follows on from your answer and from Ty's question. Our table hit a number of the things that everyone else did, but we also got stuck on politics. And we got the Mark Descent problem. The answer to overpopulation and not enough food is actually there is enough food. That it's a problem of distribution. And so then we all looked at each other and said, so, who's the economist, who's the political scientist, who's the interdisciplinary in a way that's pretty frightening. My question to you is, what institutional features in science or in this work give you the opportunity to collaborate with people who are working on these other social, political, cultural, etc.? We all can intellectually realize, yeah, that's important, but what does NSERC make you do? Or, how does it go? We've got an applied emphasis, I'm delighted to see it's there, and I hope he's more than the 20% that you have to have on the NSERC. It does keep a lot of us social sciences alive, and we appreciate the But that's the question. What institutional organizations do you see or do you think we need to build a conversation that can provide an accessible and helpful conversation?
Yeah. You know, I would say that we have disincentives. Like even having this conversation tonight, like I was worried that I might get yelled at and driven out of the room, and there's not much of incentive, there's no incentive. Now, so how do you put incentives for people to communicate more and to consider the policy ramifications, for scientists to talk with social scientists? I do think there's a lot of little mechanisms that we could put in place in the states to get an NSF grant. Now you have to have a segment about public outreach and communication. We don't do that. My answer, I can never speak to anybody but another scientist. No, for the genome.
So I can say there is one funding agency in Canada that does require that very explicitly, and that is Genome Canada on large-scale projects. And on those large-scale projects, there's a category called GELS, genetics, ethics, economics, environment, legal, social aspects of the work. And so with that research, you are required to have a team of social scientists who are doing research related to your project, looking at policy implications and a whole host of other things, but also looking, so they've got research running, but they're also involved in the delivery of applications and the considerations around those. And I have to say that I think, in my opinion, since that started, it started out as a very uneasy partnership where the social scientists were really off to the side and very much independent. And they have forced much closer work. And I think that closeness has bred much better work and has made that collaboration much easier. And so I know I've benefited hugely as a biologist from the social science teams that we've been working with on our stuff. And I mean, the ethical implications of what we're working on pale in comparison to the topics that we're like talking about gene drive in mosquitoes, for example. But we still have those conversations, and and we've had a lot of outreach around the work that we're doing. So,
Henry, then Richard. Thanks again so much for doing this, all of you. I'm just going to bring something that actually came up at the table, which was I worry less about eliminating species. Because actually, ethically, you're going, that's the kind of start. There's no return. Actually, let's use gene drive to fix, create, resistance, fix problems, save species. Because in some sense, that's the one which is going to be much harder to see the downsides to. So, can we save? You brought up chestnut trees, but if suddenly, in that context of species and diversity loss, it's like, can we use gene drive to bring back something, or to have this one resist pests that's almost wiped it out? It seems to me it's going to be much harder in an ethical debate to say, yeah, it's already in danger. Let's use this technology. Let's use this tool. And so I guess my question is that actually maybe ethically we shouldn't be worrying about should we wipe out mosquitoes, should we wipe out wolves, which seems to actually have an easier answer somewhat. It's this other side in which all the road to hell is paved with good intentions, saving species, creating resistances to improving species.
Do you want to share that?
Well, I think the example that you just referred to was the chestnut blight. So American chestnut has been absolutely decimated and is headed to extinction as a result of a disease that was introduced from Asia. And this formerly incredibly abundant species, most abundant species in eastern hardwood forests, is just almost gone. There's no resistance. And so geneticists have been working on hybridizing with Chinese chestnut and then back crossing. So you're trying to just move that one little segment in, but it's a lot faster with sunflowers than it is with trees, where you've got to wait years before they mature and fruit and everything else. So they are using a genetic modification, genetic engineering to just stick that single gene in from Chinese chestnut. It's worked very well. The trees seem to be doing well. They're out in tests, and it becomes this very interesting case study. If you were to use, this isn't a case for gene drive, but if you were to use CRISPR, it would be much more efficient in getting to that end point. But the other case we brought up was with the ash, was American ash, where we've got this emerald ash borer that's been introduced. And ash trees are incredibly common street trees. I can't remember how many million there are in Toronto that are now dead, but it is millions of trees. And in the Greater Toronto area. And so the question was, if you use gene drive with the insect, the emerald ash borer could use gene drive to deal with this issue. And that is, maybe that's the road to hell is paved with good intentions. And as you were comparing, is Plasmodium, let's compare Plasmodium and mosquitoes and wolves. And I would say, so the ash borer is a bigger insect. It's a prettier insect. They're quite dramatic. But they're non-native. So where does, extirpating it, is that a reasonable thing to do with gene drive? And what happens if the emerald ash borer with gene drive gets back to Asia?
Richard. I guess I just want to echo the thanks for the conversation. I'm realizing, as I'm reflecting on my experience, that I've got a significant degree of discomfort as we have the conversation and there's an implicit search for an answer. I think my discomfort probably arises from the fact that inherent in the search for an answer, I think, is a search for control over outcome. And I think where I'm arriving at is just the realization that I have no faith that we can control for an outcome. What I'm finding, though, is that as I'm listening to everybody here, I'm finding a great sense of comfort in recognizing the complexity of the situation. I can tolerate a bad outcome, given the circumstances. I can tolerate the bad outcome if we have the kind of conversation I'm hearing here, which is that the decision, right or wrong, good or bad, whatever that means, is going to come from a conversation which is enriched by a set of values such as non-human centric, sort of a valuing of life in a broader sense, sort of looking at the questions of power and who gets to decide. I don't need a right answer. As long as I know we can have a conversation this way about those sort of things and include that in the search for it, I think that's great. And so you've given me that a lot just from that. So I think that's really great.
Got a question over there.
Yes. Now the genomics tool is so powerful, particularly the gene editing and the gene drive, that can work as powerful as I would think it's a biological bomb, nuclear bomb. So the difference is it's so easy to achieve. So the normal laboratory can do it. Not like a physical bomb, nuclear bomb, the North Korea is doing. So even one time it makes the whole world upset. If this kind of technology can be achieved in thousands of laboratories, so what would be the control? So now we are talking about the content. If this is going to happen in thousands of laboratories, I can hardly imagine the control in the future.
A good point. I think that in the States, a lot of CRISPR research is funded by the Department of Defense. The potential use as a biological weapon is there. I think what you so now what could be done? What kind of reversals could be done? Can we mobilize scientists to do a reversal? So you're driving in an element that is devastating a crop or something that a peoples depend on or an environment depends on. Can we get in fast enough to send in something that will reverse that, will drive through the wild type, the previous allele? But we scientists do not know the answers to these things, but I think the more brains that we are saying what are the possibilities and what are the things that could go wrong, we need to think about them and discuss them. Yeah.
I think that's a question for our ethicists.
Well, don't dump that one on me. I don't know. I mean, that's an interesting question. Whether, I mean, to me, that's a question that's a number of, say, hypothetical assumptions down the road. One would be that this technology, does turn out to be, in the long term, very effective for reducing pests and other things that cause, death to humans. That in the long term, this would result in a higher rate of population growth. Not clear that that's actually the case, right? Because I mean, one thing that can happen when, say, more of your children die, you just might have more children. Right? So it might be, and people become wealthier. It's not clear that actually usually, typically they have fewer children, right? When like women, and one of the key things to the birth rate is like how much education women get, right? So if, for example, the effect of less people dying of malaria or other kinds of infectious diseases, that there's less economic loss. You lose less breadwinners, for example, in families, then families have more wealth, more women go to school, could be the birth rate goes down. So it's not, I mean, I think the connection between people dying from infectious disease and the population growing is not, is complex. Even on the assumption that the kinds of technologies we're talking about would turn out in the long run to be really effective for that. So I think it's interesting to think about a number of different scenarios too with this, like in getting back with a number of different purposes for which this could be used. For controlling pests, I mean, would it work in the short run? I mean, we're just thinking about, we've been talking like the risks. Would it work in the long run? Those are two different questions. It might be it would work in the short run, like DDT did, right, on mosquitoes. But 50 years later, it wouldn't be effective anymore, right? That's a possibility, right? And it could be, that might be enough to make it worth it for some things, right? But on the other hand, that would have a different scenario for what's the long-term effects going to be. And yeah, so.
Just to reiterate what Danielle said, I was uncomfortable with the question, but I do think that there are many levers for many of these problems, and the population lever, their education and access to family planning are levers I'm more comfortable with than disease.
Actually, Stefan, this will be the last question.
Just very quickly, my question is sort of related to things that have already been asked, but just thinking about the questions sort of framed for the panel discussion and for your talk, Dr. Otto, who gets to decide, and then the whole question of tackling the great challenges facing the planet. What I'm just wondering, I'd be interested in hearing the thoughts of the panel on this, to the extent that the very global systems of inequality, where not everybody in the world is sort of impacted in the same ways by things like global consumption of resources, things like climate change, and so forth. I just wonder to what extent the question of who gets to decide is already decided from the get-go by these global systems of or structures. I'm not quite sure the right word to use, but these sort of global disparities.
I was hoping that question would evoke the inequalities that exist right now already in who gets to decide, and that I think we should be countering that. So, for example, decisions about malaria, that's not transmitted in Canada. The costs and the benefits are elsewhere. And so how is that decision going to happen? Emily, did you want to? I think maybe if we could go around with some final thoughts. I think the answer about who gets to decide is maybe not the same as who is deciding. and that we should be trying to narrow that discrepancy.
Yeah, I agree for sure. And it definitely, weighs on me personally, thinking that, okay, I'm in one of these countries that's privileged with maybe more of a say. Like we're actually, here having a conversation tonight, whereas in some parts of the world that's never going to happen, But when I think of, so my husband was born in Zimbabwe and grew up there, which is a malaria country, and just thinking, that's where they need this, but are people there ever going to benefit from these technologies? They have so many other issues of just basic survival that they're contending with that I almost feel kind of like a responsibility living in Canada, even though it doesn't affect us, to start thinking about how we could make these things accessible for underprivileged nations. And I don't know how you do it, but and certainly malaria is just one issue among many. But I almost feel like because I get to be part of the conversation. Part of the conversation should also be how we bring these things to other parts of the world.
You then have an obligation.
Yeah, I feel a bit of obligation. Yeah. What about you?
Yeah, so I guess one thing to point out is that, and I think this kind of maybe is related to some of the questions that were asked earlier, that there's a, going back 30 or some years now, there's a tradition in a social science literature looking at ways of getting public participation into science. And there's a whole bunch of different formats and ways this can be done. And then there are people right here at UBC that are involved in projects and related to Genome Canada, for example, that do this kind of work. And so it isn't as if this kind of thing needs to be created from whole cloth. I mean, there are people who are experts at doing this kind of thing, and there are established models and all kinds of clever ways you can think about doing it. And so for example, you might have instead of to back up, I mean, obviously trying to educate everybody or get everybody to have some huge discussion about this issue isn't feasible, but you might have, say, deliberative mini publics, right, who are convened and are selected, and then of course a lot of questions about how do you recruit, which people are involved, how do you decide, on what basis, and some of my own research is related to those kinds of questions. But so I just want to, I know some of the beginning part of Sally's talk was not the most sort of hopeful kind of thing. But in terms of how do we get people involved, it's not like we don't know how to do it, right? Or I mean, there's obviously hard problems related to it. It's not like we're just completely naive and uninformed about how to do these kinds of processes. And they're out there, and there might be places where it's harder to do this than others. But I mean, there might be practical obstacles. But to me, it's not like this is something that's unknown how to do. I mean, in some ways, it might be the biggest obstacles. Might be the question that was asked where, well, there's certain people who are going to decide because they have the power to decide, and maybe that's ultimately, I don't know, like with climate change, the kind of thing that makes you depressed. But that's a different story. So, but the point is to say that, yeah, I mean, this is something that people know stuff about, and so there are ways to address these. How do we get input from the public on these kinds of issues?
So I'd like to just add a little more hopeful note. And I want to jump to the example about agriculture. And so if we look at the genetic engineering revolution and a lot of the breeding work underlying the Green Revolution and everything else, there was a lot of focus on major industrial crops, major industrial agricultural crops, corn and soy, and there's a lot of crops that are absolutely critical to people in other parts of the world that are not those crops and didn't receive a lot of attention, not a lot of breeding attention, advances in disease resistance, everything else. And I think one of the things that's happened, and the just realm of genomics is part of this, and CRISPR is going to contribute to this in a big way, is that really the ability to catch up, to take what are called the orphan crops and accelerate breeding to increase productivity under conditions of drought or salinity or heat stress, There's a lot of opportunity there, and I think that if it's being developed by or with the people that depend on those crops, I think there's a lot of opportunity. I know the Gates Foundation has been pouring huge amounts of money into this area and working on crops like chickpea and sorghum and cassava and various other things. So in some ways, these big technological advances deliver these tools into the hands of many instead of the hands of few.
Thank you for ending on a positive note to counter my very negative starting notes. I really appreciate the comments and the discussion from the audience. Really, every question was fantastic, and the conversation was really good. And I do hope that everybody goes and talks to this many people, and maybe by continuing on these conversations. And I know from having this panel, I've already met other people on campus that are grappling with these issues. And so I do think that there will be more movement on this within UBC, and hopefully we can spread it.
Please join me in thanking our panelists.